Hello, everyone. I'm Charles Mulligan, Deputy Director of the St. Jude Comprehensive Cancer Centre. It's my great pleasure to welcome you to the 12th in our current series of the Science of Childhood Cancer. And I'd also like to mention the next three talks in our series. Next week, Xiaomin Wang will speak. Xiaomin is of St. Jude, and he will discuss genetic and epigenetic approaches to understand chronic health conditions in childhood cancer survivors. And then the final two talks are from external speakers. So on April the 29th, we welcome Kim Stegmeyer of the Broad, who will present a first-generation pediatric cancer dependency map. And on May the 6th, the last speaker in our series is Sigal Kadok, who will present structure and function of switch-NIF chromatin remodeling complexes in human cancer. So be sure to join us again for those three fantastic sounding talks. And finally, I thank all of you for joining. The response to these series has been truly phenomenal, and it's been a great privilege to showcase great science from St. Jude and beyond. So now it's my honour and pleasure to introduce today's speaker, Madan Babu. I'm very glad to now welcome Madan as a colleague and collaborator. Madan and his group joined St. Jude virtually in July of last year and just recently arrived in Memphis. And he was just telling me beforehand how he had a truly Memphian welcome with a bolt of lightning striking his house just as he was arriving in the city. So with that propitious start, he's now a member of the Department of Structural Biology and the Endowed Chair in Biological Data Science and Director of the Centre for Data-Driven Drug Discovery. Prior to that, he had a truly distinguished career. He was tenured program leader at the MRC Laboratory of Molecular Biology, and he has diverse and broad and deep research interests that include the use of systems biology and large-scale data mining approaches to understand biological systems, cancer, and drug development. Among his impressive and many honours are being recipient of the EMBO Gold Medal in 2019. He has a specific interest in the regulation of G protein-coupled receptor signalling and the roles of alternate RNA splicing in health and disease. So it's timely to welcome Madan to the Science of Childhood Cancer for his talk, Data Science Approaches for GPCR Signalling and Implications for Drug Discovery. So thank you, Madan, and over to you.
Thank you very much for the kind and generous introduction, Charles, and good afternoon, everyone. It's a real pleasure for me to be here virtually, and I'd again like to thank Charles Mulligan and Charlie Roberts for inviting me to present today. And what I'd like to do in the next 45 minutes or so is talk to you about some of our work on how we use data science approaches to understand GPCR signaling and discuss the implications for drug discovery and development. All right, so technological advancements have allowed us to generate a huge amount of information describing biological entities and processes at many different scales and resolution. Now, this presents us with unique opportunities to use computational methods to gain fundamental insights and discover new scientific principles. So the current estimates, according to the National Institute of Health and the European Bioinformatics Institute, is that we have access to over 150 petabytes of processed biological data. Now, given this wealth of data, what drives us scientifically is to find out how we could discover new biological insights and knowledge from these large volumes of data. So the mission of my group is to develop data science approaches to make discoveries and reveal fundamental principles of living systems, and the emphasis on understanding human biology and pediatric catastrophic diseases. Now, to achieve this objective, we investigate biological systems at three distinct levels of complexity: the atomic level, the cellular level, and the population level. And we bridge these by investigating the impact of mutations across these different scales. Now, to this end, we develop interdisciplinary computational and experimental approaches that generate, integrate, mine, and analyze disparate data sets. And these include data describing sequence, structure, expression, and networks of interactions, and many more. So our approach to understanding human biology through data science approach is to first define groups of proteins that are medically relevant or have unusual structural properties. And we bridge, again, the understanding of these proteins across the different scales to gain insights. And the major scientific directions of my group so far have been on G protein-coupled receptor signaling in human physiology and disease, as well as to understand the roles and functions of disordered protein regions in human biology and disease. And in today's presentation, I'll largely focus on some of our work on GPCRs and will not be talking about our work on disordered proteins. So this is going to be the outline of my presentation today. First, I'll give a brief introduction to G protein-coupled receptors, and then the main part of my talk will have two different projects that we finished. One is on understanding the pharmacogenomic landscape of GPCR drug targets, and the other one is to understand the role of GPCR isoforms and how they could make signaling more complex in different human tissues. And I'll end by discussing some of the future directions that we are pursuing here at St. Jude. All right, so what are GPCRs? So GPCRs, or G protein-coupled receptors, are the largest family of membrane protein receptors in the human genome. There are more than 800 different genes that code for GPCR genes in the human genome. And each one of these receptors participate in diverse physiological processes ranging from sensory responses such as vision, taste, and smell to those regulating behavior, the immune system, the cardiac system, among others. And in this manner, GPCRs govern virtually every aspect of human physiology. A number of different GPCRs have been implicated in diverse cancers, ranging from its role in angiogenesis and metastasis to applications in cancer immunology. Some classic examples include class F receptors such as frizzled and sonic hedgehog, and several of the chemokine receptors. Now, given their role in modulating physiological processes and their presence on the cell membrane, it is not surprising that they serve as attractive drug targets. Currently, over 1/3 of all FDA-approved drugs target about 108 different GPCRs, and over 300 different agents that are at various stages of clinical trials target an additional 66 different GPCRs to treat diseases and disorders such as obesity, diabetes, asthma, among others. Now, given the medical relevance of GPCRs, there's been enormous progress in structure determination in the last couple of decades. And we now have access to more than 500 different structures of about 90 different receptors. Thanks also to the advancements in electron microscopy in general, we now have complexes of GPCRs with many different adapter proteins, such as G proteins, RAMs, and arrestin. Each new structure has provided unprecedented insights into how these receptors function. And in our group, we were interested in discovering general principles and common mechanisms by which this protein family functions. So how can we draw insights from all these data? Now, when you have two structures, it is relatively straightforward to understand what the structural changes are. But when you have over 500 different structures, it becomes non-trivial to be able to objectively identify which changes are relevant and which changes are less consequential for function. Now, to address this problem in our group, we have developed network-based approaches and data science approaches that represents protein structures as networks of interactions. And we have applied data science techniques to investigate what are the relevant biology that one could glean from looking at conformational changes in these structures. So what have we done in this space? So primarily through the work of two extremely talented PhD students at the time, A.J. Venkatakrishnan and Tilman Flock, we have developed computational methods to investigate protein structures as networks and integrated them with diverse genome-scale datasets to provide mechanistic insights into GPCR signaling. All the way from understanding principles of ligand recognition and how ligand binding leads to receptor activation, to understand the code by which GPCR selectively recognize G proteins, as well as the allosteric network of activation, eventually resulting in GDP release. And through the work of another summer student, Andrea Center, we were also able to understand the interaction between GPCRs and arrestin. Now, to facilitate network analysis of protein structures for any protein family, we also developed what's called the Protein Contact Atlas that allows scientists to analyze structures as networks and derive insights into how changes associated with protein structures can contribute to function. Now, with this introduction, I'd like to move on to the first part of my talk, where I'll discuss some of our work on pharmacogenomic landscape of GPCR drug targets. Now, having focused on analyzing sequences and structures and having contributed to understanding some mechanistic insights into this large family of proteins, we became interested in applying this knowledge to a problem in human health, and therefore we focused on pharmacogenomics. Now, it has been estimated that adverse drug reaction can cost up to $30 billion just in the US alone. And despite the importance of GPCRs in human physiology and as drug targets, one thing caught our attention. There was no variant information that is currently included in labeling information of drugs. Now, is this because GPCRs are not variable in the human population, or because we have not looked into this issue carefully, was not known at the time of the study, and this got us interested in this problem. So again, with the help of another very talented PhD student, Alex Hauser, who visited us from David Glorium's group, we became interested in this problem and addressed the following questions. So given that GPCRs are attractive drug targets, how polymorphic are the drug targets in the human population? Do individuals with variant receptors respond differently to drugs? And what are the associated economic burden due to variation in GPCR drug targets? And addressing these questions have implications for personalized medicine, which I'll touch upon a little bit later in my talk. So more specifically, to address the first question, we could either look at an individual and ask how many different polymorphisms do they carry in different receptors, or we could have a receptor-centric view and look at which receptor is the most variable or least variable in the human population. So to address some of these questions in terms of how variable the GPCRs are in one particular individual, we analyzed the data on FDA-approved drugs and which GPCRs that they target. We then integrated information on genome variation from the 1000 Genomes Project, which provided information about 80 million SNPs from about 2,500 healthy individuals from different parts of the world. Now, we find that on average, each individual in this data set carried about 68 missense mutations in about 1/3 of known GPCR drug targets. Now, interestingly, on average, again, about 8 variants have been previously associated with altered drug response from other population-wide association studies. For instance, the A307T polymorphism, the follicle-stimulating hormone receptor, is prevalent in women who develop polycystic ovarian syndrome and is also associated with higher responsiveness to exogenous FSH. Now, similarly, the G9S polymorphism has been linked to an increased risk of gastrointestinal toxicity when treated with levodopa in people with Parkinson's disease. To also assess how often new mutations arise in the human population, we investigated the de novo mutation database, which contains genome sequence information of father, mother, offspring trios. And an analysis of this data revealed that mutations in GPCR drug targets arise in 1 out of every 300 newborn children. And what these observations suggest is that there is extensive variation in GPCRs, and new mutations do continue to emerge in the human population, and that this may potentially contribute to an as yet underappreciated variability in drug responsiveness. So we then focused on our attention on individual receptors, asking which receptors vary more or vary less in the human population. And to address this, we had access to a much larger data set. At the time, the Exome Aggregation Consortium, or the EXACT data set, provided information or aggregated mutation information from over 60,000 individuals in the human population. We also analyzed copy number variation, gene duplication, and gene deletion data, but in the interest of time, I will largely focus on missense variation here. Now, we find that a total of about 14,000 missense mutations occur in about these 108 GPCR drug targets, with several rare alleles as well as a few common alleles. Now, some of these receptors show more variation than the others. An example of the most variable receptor included the mu opioid receptor, which I will talk about a little bit later in the talk, and the dopamine receptor D5. So it does look like GPCRs that are targeted by common drugs do show variation in the human population, meaning that different people have slightly different versions of the same receptor. Now, by integrating information on the extent of variability of GPCR drug target with FDA-approved drugs, we find that several of the highly polymorphic receptors are also targeted by a large number of approved drugs, like the mu opioid receptor being targeted by more than 30 different FDA-approved drugs, which is also highly variable in the human population. So what these observations collectively suggest is that the extensive genetic variation in GPCRs that are targeted by these drugs may again contribute to this underappreciated variability in drug responsiveness in the human population. All right, so far what I've tried to show you is that GPCR drug targets are variable. However, the more important question is whether the individuals who have this variation likely to respond differently to drugs, and whether these variations really occur in functionally important or relevant sites. Now, to address this, we analyzed all available structures of GPCRs at the time using Protein Contact Atlas that we developed in a group, as well as integrate multiple large-scale data sets describing post-translation modifications from mass spec experiments, as well as identify functionally critical positions and integrated them with the genome variation data. Just to elaborate on this a bit more, as I mentioned, over the years, through the work of the two PhD students at the time in our group, we were able to identify positions that are critical for maintaining the GPCR fold, residues important for ligand binding, receptor activation, and how exactly they are able to bind to G proteins, as well as activate G protein upon binding. So by mapping all this polymorphism data, we find that over 2,000 of the mutations fall within known functional sites. So roughly about 14% of the missense mutations fall directly in one of these sites. Now, what is the likely impact of these mutations? So we find some of the mutations fall within drug binding pockets, which are shown in green here. And there are other mutations that fall within functional sites, such as post-translation modification sites, such as glycosylation or phosphorylation and ubiquitination sites. And all of these could potentially influence receptor expression level, as well as desensitization and internalization of these receptors, meaning how long they stay on the membrane, how much of the receptors make it to the membrane, and how quickly they can actually be removed from the membrane. A few others also fall in the vicinity of where exactly they bind to effectors, such as G proteins and arrestin. And all of this essentially suggests that individuals carrying some of these polymorphisms may respond differently to drugs, either due to ineffective drug binding or altered receptor expression level, or binding to effectors differently. Now, this slide shows all currently available structures of GPCRs in complex with an FDA-approved drug. And just to highlight and give you one example, I'll focus on the CCR5 receptor, which is the target of the anti-HIV drug, maraviroc. Now, 8 of the 9 positions that contact maraviroc are polymorphic in the human population, suggesting again that individuals with these mutations in drug binding pocket may have altered efficacy when treated with this drug. All right, so taken together, what these findings highlight is that there is widespread diversity and that this can potentially impact GPCR drug targets with potential implications for drug binding, receptor activation, and downstream signaling. Now, the next question that we asked was whether there is actual real evidence that the variant receptors do bind and respond differently to drugs. And for this, we focused on the mu opioid receptor, which is one of the most polymorphic receptors in the human population that I mentioned early on in my talk. And as we all know, opioid analgesics are the one of the most effective pain drugs, with over 25 different drugs targeting them, such as morphine, codeine, and fentanyl. And while pain relief is a desired effect, a major side effect is that individuals also get addicted to drugs, and opioid addiction is a huge societal burden. And overdose is also a major dose of opioid-related deaths. To investigate the effect of some of these polymorphisms, we collaborated with a group of Kirill Martimianov at Scripps, Florida, to develop an assay and test the impact of some of these mutations. Now, in this assay, in this slide, the wild-type receptor structure is shown on the left. And we first developed a bread-based assay that could reliably discriminate between the actions of 3 different pharmacological agents. Morphine, which is an agonist and used for pain relief, behaves the same way when treated with endomorphine, which is the endogenous ligand for this receptor. Buprenorphine, which is a partial agonist and is used for de-addiction, shows a partial response. And finally, naloxone is an antagonist and is usually given as an antidote when patients are hospitalized due to opioid overdose. And in our assays, as expected, this shuts down the response. Now, we found that several receptor variants could influence these drug responses. And here I will focus on 3 specific polymorphisms near the ligand binding pocket that are shown in red, blue, and green. Now, when treated with the endogenous ligand, all these receptors with these polymorphisms behave in a similar manner, with the red receptor or red variant showing slightly reduced response, suggesting that individuals with this variant are unlikely to respond effectively to morphine for pain relief. Now, intriguingly, when treated with buprenorphine, the drug that should provide partial response and help with de-addiction, we find that the red variant shows no response, whereas the green and the blue variant shows much higher response than the wild-type sequence. In other words, a partial agonist is behaving like a full agonist at these variant receptors. Now, this suggests that if individuals who have this polymorphisms are treated with or for de-addiction with this drug, they are less likely to respond effectively as they're getting what they essentially want. And the most serious situation happens when treated with naloxone, where the wild-type and the red variants behave as expected, whereas the green and the blue variants show a full response or a partial response, depending on which mutation you look at. And this means that if individuals with this mutation are hospitalized for opioid overdose and treated with the antidote, rather than shutting down the response just keeps going on, suggesting that it could potentially lead to more serious adverse reaction. So the key point that I wanted to highlight is that polymorphisms need not always result in loss of function, but they could actually show gain of function effects. And more importantly, the effect of the mutations on drug response may not be obvious by simply studying the response of the mutant receptor to the natural ligand. All right, so putting all these observations so far together, what these highlight is that receptor variants have the potential to lead to unintended consequences when treated with drug. It could be an adverse reaction or an altered reaction or ineffective response. Now, this takes me to the last question in this section, which is what is the likely economic burden that is associated with variation in GPCR drug targets? Now, to address this question, we made use of the drug sales data from the Open Prescribing Project, which provides information about all the GP prescription through the National Health Service in the UK and the cost of making these drugs. We also used the British National Formulary Classification to group the drugs for our study. Now, based on the data from about 279 drugs, the NHS spent about £1.7 billion on drug costs on GPCR targeting drugs in 2016 alone. And we modeled all this data and inferred the rough economic impact due to target variability. So in the interest of time, I won't be going into the details, but the estimates range from £14 million annually to about £500 million, depending on how conservative you are, which is on the lower end of the spectrum, analyzing only 14 receptors to all the known receptors, where the number goes up significantly higher, suggesting that investing efforts in understanding and actively incorporating the effect of polymorphisms on drug response can potentially have an impact on unaccounted expenditure on public health. Now, towards the goal of advancing the field of personalized medicine and fuel pharmacogenomics research, we have also created a comprehensive resource within the G protein coupled receptor database. This is the go-to database for GPCRs that is maintained by our collaborator, David Gloriam, at Copenhagen University. And I would encourage you to visit if this is of interest. And the website can be accessed through www.gpcrdb.org. Now, those interested in specific receptors or drugs or drug classes can obtain all the information that I described here through this online interactive resource. All right, so in summary, what I've showed here for this part of the talk is that GPCRs that are targeted by drugs do show variation in the human population, and that such variation occurs in functionally important sites and may result in altered drug response. And understanding GPCR genetic variation may potentially help alleviate a global economic burden. Now, this takes me to the last section of this part, where I discuss some of the implications of this work. Now, what I've shown in this part of the talk is that we are now in a position to bridge biological information across different scales, from those describing atomic level details of the structure of receptors, all the way up to data describing population scale differences in genome sequences. And now through data integration, one could potentially assess the prevalence, functional, as well as the economic impact of GPCR drug target variation and variable drug response. So one obvious question which does come to mind is that why do we find such variation in GPCR drug targets? Now, a bit like endomorphine and the mu-opioid receptor, we think that most polymorphisms may not have a significant effect on the endogenous ligand, or only have moderate effect and may have physiologically acceptable range, leading to minor phenotypic variation in the physiology that we see in the human population. However, they only manifest when treated with artificial chemicals such as drugs. So in other words, a substantial fraction of the population might carry variant receptors and remain healthy and pass on the genes. However, they have the potential to display differences in drug response if and when they are treated with a drug, probably much later in their life, much greater than and past their age of reproductive fitness. So I'd like to emphasize that there are several factors that one needs to consider to interpret some of the results that I presented here. And to really gain deeper insights, one has to think about pharmacogenomic effects of these variant receptors in the context of the fact that we are all diploids, so which means the effect of heterozygosity has to be considered, allele-specific expression, importantly, drug metabolism and kinetics. And here the Department of Pharmacology has really pioneered some very important concepts in pharmacogenetics and pharmacogenomics, establishing these concepts directly to patients here at St. Jude. Effect of non-coding mutations, expression variation, differences due to gender and age are all some of these things. Nevertheless, with all these considerations in mind, some of the results presented here do provide an interesting glimpse of the extent of variation within the coding regions of GPCR drug targets. And the general idea is that taking this further, one could use all this information to move away from the current model, which is one treatment fits all, to the future where we can stratify patients based on their genotype, and also to be able to either alter dose or suggest alternative medications to maximize effective as well as minimize adverse drug reactions. So what I want to also highlight is that on the computational front, that the multi-scale data integration framework that we have developed here can actually be used to study other drug targets such as ion channels, nuclear receptors, kinases, and so on. And a number of members in our group are looking at some of these drug families. Now, it can also be used for pharmacovigilance investigations by regulatory bodies to see if drugs are responding as expected, as most drugs were approved way before explicitly considering genetic variation data. And most importantly, it could be used to stratify patients entering clinical trials to maximize success and clinical outcome. Now, personally, one exciting possibility is that it also means that one could look back at failed drugs to see whether it would still work for a certain subpopulation. And I think these are some interesting avenues that one could start thinking about more closely. All right, so at this point, I'll switch sections and we'll move on to the second major part of my talk, which is on GPCR isoforms and signaling complexity. So this is a work that was spearheaded by a very talented Marie Curie postdoctoral fellow in my group, Dr. Maria Marti. And here what we were interested in was to understand tissue-specific differences in GPCR signaling responses by investigating transcriptomic data from over 30 different human tissues. Now, as I mentioned, there are 800 different GPCRs. Each one of these receptors can detect and respond to diverse ligands, ranging from a single photon in the case of rhodopsin, all the way to small proteins and peptides, like in the case of chemokine receptors. So these membrane protein receptors can regulate diverse signaling pathways inside the cell upon binding of the ligand. There are 16 different heterotrimeric G proteins, arrestins, RAMs, and each one of these can then go on to trigger multiple different signaling cascades. Now, in humans, we have over 50 different tissues, 200 different cell types, and 37 trillion cells. So the question that we became interested in was the following. Now, given that there are so many different organ types that are made up of many different cell types and such a large number of cells in the human body, do all the cells and tissues that express a receptor respond the same way to a given ligand? If not, what are the mechanisms that can diversify such signaling responses in different tissues, and how prevalent are these mechanisms and diversification of signaling response to the same ligand? Now, understanding this is important because it does tell us about basic biology, as well as giving insights into variability in drug response. So we focused on one molecular mechanism called alternative splicing. And as we all know, alternative splicing is a molecular mechanism that results in the generation of multiple transcript isoforms. And if you include or exclude exons differently, you can potentially alter the sequence of the protein that eventually are coded by some of these open reading frames. Now, advances in next-gen sequencing have shown that over 90% of the human genes undergo alternative splicing, and nearly half of the isoforms have tissue-specific distribution. So how exactly this affects GPCR signaling on a system level was currently unknown when we embarked on this study. So the questions that we became interested in were the following. How many GPCRs have more than one isoform? And how exactly do they differ in their structure and function? Now, how does their expression contribute to diversification of the signaling responses in different tissues? And importantly, what are the implications for drug response as well as for developing new drugs. Now, to address this, we made use of one of the largest resources that is currently available called the GTEx resource. And this has information about isoforms as well as expression levels from 30 different tissues and over 700 different individuals, and integrating that information with the G protein coupled receptor database and developing a computational pipeline, we find that over 154 of the receptors, roughly about 40% of all GPCRs, have 2 or more different isoforms. And we could detect about 730 different isoforms that are expressed in these different tissues in this data set. Now, we also developed computational methods to ask where exactly and which part on the structure many of these isoforms affect the sequences or alter their conformations. And we find that a majority of them end up affecting the N-terminal region or the C-terminal region. We do find a large number of receptors that create truncated products. They affect all the different classes of GPCRs for the GPCR aficionados. Now, interestingly, the fact that they affect N-terminus and C-terminus essentially means that they can have directly an impact on ligand recognition binding, as well as kinetics of internalization, receptor expression, as well as homeostasis and turnover on the membrane. And all of these become very important criteria and considerations that I'd like to highlight in the next few slides. Now, we also integrated published mass spec data sets describing where exactly post-translation modifications occur. And we do find examples where certain C-terminal tails that have phosphorylation sites are differentially included or excluded in these receptors. And phosphorylation of the C-terminal tail is very important for it to be able to recognize arrestin and mediate receptor internalization. So when a ligand binds, how long it stays dictates how much it can signal through the cell. And phosphorylation and internalization of the receptor ensures that the signal is shut off. So we do have receptors where the C-terminal tail with the phosphorylation site are affected, suggesting that there could be subpopulations of receptors that may respond to different times to the same ligand. So we also mine the literature, and we find instances of isoforms that have been extensively biochemically characterized, specific isoforms that have been extensively biochemically characterized by a number of different research groups. And we identified examples where the N-terminus that show an alternative sequence can affect ligand binding, or the C-terminus that have an altered sequence can affect G protein binding as well as internalization, or trafficking if it affects post-translation modification sites, as well as truncated versions that can influence the expression of the wild type sequence in the receptor because it can act as a dominant negative version of these receptors and retain them in the ER without allowing them to go all the way up to the membrane. So again, by analyzing this data, we find evidence for a large number of previously uncharacterized isoforms that have variation in these exact regions where some of these receptors have been very well characterized in the literature. And together, we could arrive at the following conclusions, which is that most isoforms do affect hotspots for ligand binding and receptor coupling to different signal transducers, and that characterization of these regions that are affected can suggest the most likely functional impact of these unstudied GPCR isoforms. So the key message is that isoform structural variation can diversify receptor function. So the next question is, where are they expressed? Are they expressed in combination? Are they expressed in low levels and so on? So how exactly does the expression contribute to diversification of signaling response? Now, to address this, what we did was to come up with a simple metric that allows us to identify the complexity of co-expression. So I just want to walk you through this toy example. On the x-axis are the different tissues, and on the y-axis are the different isoforms. So here's an example of a receptor with 2 different alternative isoforms. And you can see that in this toy example, all the isoforms are expressed in all the tissues equally, suggesting that there is no variation in co-expression, and thus the tissue expression signature is 1. Now, here's an example of a receptor with 2 different isoforms, but you can see that different tissues express different combinations. And in this case, you have a tissue expression signature of 4. There are 4 different patterns of combinations, again, suggesting that for the same ligand, you can expect slightly different responses in the different tissues because you just don't have the reference isoform, but you also have alternative isoforms that are co-expressed. Now, this is a real-life example involving the cannabinoid receptor as well as the CD97 receptor, where you have one receptor with two isoforms, and you can see different tissues express different combinations. So the expression signature is 4, whereas in the case of CD97, all the different isoforms are expressed in all cell types, suggesting that there is not a big difference in the complexity of a signaling response. So when we looked at all these different tissue expression signatures for each one of these receptors, we do find that there is variation in this tissue expression signature, suggesting that there is a complexity in signaling response in these different tissues that could be contributed by co-expression of these alternative sequences. So to address whether expression of these isoform combinations can contribute to signaling bias, we focused on the cannabinoid receptor 1, which has 3 different versions in the N-terminus, and this is exactly the region where the receptor contacts the chemokine ligand. And what we could find through collaborations with Manoj Puthenveedu and Stephanie Crilly at University of Michigan, we were able to find that co-expression of different isoform combinations can drive distinct cell signaling states. And what this means is that if we are not aware that an isoform is expressed in a tissue or a cell line of interest, the same ligand can give rise to different signaling responses. So if there are 2 labs that characterize the expression of a wild type receptor, you can get 2 different results because an isoform may be co-expressed in these cells, and we may not know that these isoforms are actually expressed in these cells. And this can have implications for developing drugs, developing models for drugs, and also understanding tissue-specific responses. And we also looked at the gastric inhibitory peptide receptor, which is another receptor that showed an alteration in the C-terminus. And here, again, through collaboration with Graham Latz at the University of Cambridge, we could show that expression of different isoform combinations can result in signaling bias. And this is a very interesting and important point, because signaling bias essentially means that when a receptor binds to a ligand, you can either activate G protein or get internalized through arrestin, and you can have arrestin-mediated pathways. One becomes the desired effect of a drug, the other becomes the potential side effect of the drug. Now, what we find is that if we co-express isoforms, you can naturally bias the signaling either through G protein or through arrestin, again suggesting that this can have implications for drug development, because the natural signaling state of a cell is very different in different tissues, and therefore the same drug can respond or lead to different responses in these different tissues because of co-expression of these isoforms. So the key point here is that all tissues can express non-reference isoforms, and that receptor isoform combination may influence signaling complexity, and that such combinations can diversify such signaling responses, driving physiological signaling bias. So what do these results mean for GPCR drugs? Now, we focus specifically on drug targets again, and we find that 40% of the drug targets have 2 or more isoforms. Now, this can potentially lead to complications, or may also possibly lead to new opportunities where one could think about developing isoform-specific drugs. Now, to address the latter part, we developed a computational pipeline wherein we try to ask whether there are isoforms that may represent potentially interesting drug targets because they have a region that is accessible to the ligand that is different in sequence, and they have patterns of expression in the right tissues in which you may want to have a desired drug action to happen. And we were able to identify about 42 receptors and 64 isoforms that fit this criteria, suggesting that these are likely interesting targets for future studies. Now, I'm just going to show you one example, which is the chemokine receptor CCR3, where the N-terminus is altered. And as you can see, this is exactly the region where it binds to the ligand. And you can see that the 2 different isoforms are expressed differently in some of these tissues, suggesting that one could think about developing a molecule that can target the presence or absence of this extra region in certain cell types for mediating an effect that is desired. Now, we also asked whether these isoforms really have any associated phenotypes in the human population. And to address this, we mined the largest data set that's currently available, which is a UK biobank that has information about genotypes from over 500,000 different people. And analysis of this data, which is done by Duccio Malinverni in our group, we were able to show that there are polymorphisms that are only present in isoforms but not in the reference sequence that are uniquely associated with different phenotypes. And what this essentially suggests is that there is an underappreciated potential for isoform-specific therapeutic indications that can be looked at more closely. So in summary, for this part of the talk, we find that about 40% of the drug targets that are targets with approved drugs in the market do have more than one isoforms in humans expressed. Now, there is genetic evidence that links receptor isoforms with specific phenotypes in the human population, and that the non-reference isoforms, as much as they can potentially complicate drug response, can also represent potentially novel drug targets. All against highlighting that one could think about rationally exploiting isoform-level drug selectivity to potentially identify new drug candidates. Now, as with the previous study, our collaborator, David Gloriam, who maintains the GPCR database, also maintains some of the results that we could identify from these studies. So anyone's interested in GPCR isoforms or the expression in the different human tissues, you're very welcome to visit gpcrdb.org that has all this information in one place. All right, so in summary to this part of the talk, what I've tried to highlight is that through a systematic integration of multiple large-scale data sets ranging from expression data to sequence and structure data, as well as literature data, genetic association data, and drug data, one could gain insights into the prevalence and likely functional impact as well as signaling complexity due to the expression of diverse GPCR isoforms. All of this points to a very interesting concept in our view, which is that we now need to move from a canonical view where we can imagine one receptor regulating a G protein or an arrestin to what is called as a context-specific view in GPCR signaling, where in each tissue, it is not just one receptor, but an ensemble of receptor isoforms that are co-expressed that determines and drives the cell signaling state of the cell, which means in different tissues, you can have different combinations of isoforms that are expressed. And what this essentially means is that for the same ligand, you could have tissue-specific responses that are mediated due to the combinatorial expression of some of these isoforms. So varying these isoform repertoire can result in system-specific signaling outputs, and therefore an obvious corollary is to ask whether there are diseases where these combinations are perturbed that may drive to pathological states. Now, this is something that we are interested in pursuing here at St. Jude. So in conclusion to this part of the talk, we find that GPCR isoforms do affect functional hotspots in the receptor and can diversify structure-function relationship. Now, the expression of different combinations can result in different physiological states and can drive what is called physiological signaling bias in tissues, and that such isoforms could explain potentially complex pharmacological profiles where things may work in cell assays but may not work in tissues or in like whole organisms, because one need to consider the whole complexity of isoforms and their co-expression in the different tissues that may eventually result in altered drug responses. There are a number of implications and discussions. I won't go into the details in the interest of time, but I'm happy to talk about it much later. Now, the key point here, however, I'd like to highlight is that such isoform diversity may potentially explain how and why organisms can mount different responses to the same ligand in different tissues. However, it may still lead to a coordinated homeostatic response at the whole organism level. So you can have this diversity that comes together to result in a unified physiological response. All right, so in the last 2 or 3 minutes, I'd like to just very briefly mention some of the future directions that we are pursuing here at St. Jude. Now, I just want to take a moment to highlight and hope that these 2 studies have tried to show how analyzing large amounts of information can allow us to answer fundamental questions that are of very different magnitude and kind, that is very complementary to contemporary approaches that are out there. Now, how can we take these discoveries that one can make on GPCRs and the isoforms to gain deeper molecular insights and possibly identify new compounds that can selectively target these isoforms? Now, thanks to Dr. Babis Kalurimus here, who has built an outstanding structural biology department at St. Jude with all the major structural biology methods in various core centers, what we are currently doing now is to perform integrated structural characterization of some of these polymorphisms and isoforms through the various core centers in the department. Now, I also want to say that at St. Jude, it's been just fantastic because we have what is called as the St. Jude Data Science Focus Group. Again, thanks to the initiative of Dr. Tomi Mori and Stan Pounds, we have an amazing group of data scientists from many different disciplines, all the way from epidemiology and human genetics to image processing, chemical biology, and genomics. So moving forward on the data science front, what we would like to do is to take an integrative approach to understand GPCR signaling through the lens of biological data. So specifically, as we all know, cryo-EM is leading the resolution revolution, providing atomic level insights into more and more new structures and complexes that are coming out at an unprecedented rate. Now, single-cell transcriptomics is also revolutionizing our understanding of cell types, cell states by analyzing expression data of genes in the resolution of individual cells. And the genome sequencing methods are paving the way for obtaining complete genetic information of individuals in the human population. And what we would like to do is to investigate signaling across all these different scales to better understand human biology that is governed by GPCRs. Now, beyond our work on GPCRs, we've also been focusing and bringing in the approaches that we have developed to study other medically important gene families that are of interest here at St. Jude. So we've also been focusing on kinases, nuclear receptors, as well as zinc finger proteins. All right, so in conclusion, I hope I've shown how analyzing large amounts of information can allow us to answer interesting and important questions of a very different magnitude and kind that is complementary to current approaches, and how such approaches can potentially help convert this large amount of data into knowledge. And hopefully here at St. Jude, we can see how this new knowledge can be potentially put into action in the coming years of our time here. So with this, I'd like to conclude my presentation and acknowledge several of my group members, as well as funding agencies, particularly St. Jude for supporting our research, and to the UK Medical Research Council for supporting our work over the years. I've also acknowledged many of my collaborators, co-workers, and the key people who led the project as we go along in my talk. I'd also like to thank Dr. Downing, Dr. Morgan, and Dr. Colodimus for the opportunity to establish this new Center of Excellence for Data-Driven Discovery here at St. Jude. So in this last slide, I just want to just mention that we are going to have several positions where we will be looking out for outstanding scientists who would be interested in various aspects of data science as well as experimental biology. So we'll have a number of new job openings looking for staff scientist positions for cryo-EM and mammalian protein science. And there will be upcoming data science opportunities for those interested in cancer genetics, human genetics, omic data analysis, synthetic biology, pharmacology, and molecular cell biology. So if anyone's interested, or if you know of colleagues, especially participants at this talk, please do let them know about our interest and our efforts to understand GPCR signaling and their role in cancer, especially in pediatric cancer. Thank you all for your time and attention, and I'll be happy to take any questions that you may have.
Thank you so much, Madan, for that really fascinating and astonishing sweep through the field of GPCR research. What an incredible odyssey of discovery it's been for you and your team. We're especially glad to have you here now. So again, just to everyone on the WebEx, it's question time. Please feel free to ask questions, put them in the chat box, but importantly, address them to all panelists. We will continue for a few more minutes, then conclude the formalities at the top of the hour and continue for extended Q&A after that time. So, first question from Jingwei Zhang here at St. Jude. Madan, have you evaluated whether germline polymorphisms have any effect on relative quantity of isoform expression?
That's a great question, Jinghee. We have not done any studies on non-coding variation or even mutations in intronic regions, or these non-synonymous SNPs may have an effect on expression level. Hopefully in the future, these are questions that we could look at more closely. We did try to identify EQTLs. So these are polymorphisms where you can alter the relative expression of the isoforms. We did find a few of them, but we haven't pursued them further. In another ongoing study, we've been looking at differences in expression level between males and females, as well as the effect of mutations in intronic regions. So hopefully we'll be able to address some of these questions in the coming days. Thank you.
Okay, great. So next question is from Nick Giangreco. How well are drugs able to be delivered in a tissue-specific manner, and does exploiting this isoform diversity rely on specific drug delivery?
Yeah, it's a great question because, I mean, we now need to probably think a little bit more about drugs beyond small molecules, like chemical molecules. So one could think about other mechanisms by which certain tissues that may be more accessible to other non-conventional mechanisms of drug delivery. So one idea that we've been thinking about, especially with targeting isoforms, is whether one could think about small RNAs that could now selectively target the expression of one isoform or the other. Because if some of these changes happen in what you may call regions that are not accessible, and therefore they're not on the outside of the membrane, then it becomes very difficult to target them using conventional approaches. And that's exactly where other mechanisms and methods may come in handy. So antisense oligos, like what has been developed to treat ALS and other diseases, I don't know, but those are some avenues that could be thought about. PROTACs are other class of drugs which are still small molecule based, but they're not stoichiometric. In that sense, as long as you can bring in an E3 ligase that can degrade the receptor of interest, then one could think about mechanisms like those to change the expression level of the gene. PROTACs have been actually developed against ion channels and transporters. So I'm sure someone's already developing this for GPCRs.
Next question is from Sarah Sherman. The tissue differences in isoforms is very intriguing. How much do you think these tissue signatures vary between people? And do you think these signatures could relate to disease predisposition, that is, a predisposition to addiction, for example?
Right. Thanks, Sarah. That's a really important question. So one dimension that I have not discussed today is the time dimension of an individual. So from birth to individuals who age over time, how does that tissue-specific expression even change, right? So there are data sets that have just become available. The group of Henrik Kassaman from Zurich have published data sets describing expression levels of transcripts from fetus all the way up to very old individuals. So mining those data could already give some insights into whether tissue expression signature varies over the lifetime of an individual. Now, this is an important calculation to do before addressing some of the questions that you raised, because we need to age match individuals to even understand how much variation actually happens. It cannot come purely due to developmental differences, but it should come from true genetic differences in isoform co-expression due to polymorphisms or variation. So addressing that question may require addressing one of these questions first, which is something that we hope to do in the coming months.
Okay, and there are a couple of other questions that were really examining the same thing about isoform diversity changing over time and with age. So a different question from Martine Roussel here at St. Jude. So from Martine, how can variant sequences be used in patients before treatment as an opioid variant, and how will this be deployed?
Yeah, no, I mean, that's a very important question, Martine. So one project that we initiated but we couldn't complete was with a collaborator from the MRC toxicology unit in Scotland, where they had samples from patients who died due to opioid overdose. So one idea was to go back and look at whether they ended up carrying certain polymorphisms that, if one could screen upfront, could at least flag them as individuals who may have an altered drug response. So one idea that we thought was, just like how people come with what's called a polygenic risk score, which is to look at the sum of different polymorphisms that have weak associations, but cumulatively they act as reasonably good predictors of development of a complex disease. So we were thinking that maybe one could think about coming up with what's called a polygenic or pharmacogenomic risk score that would allow us to identify whether there are individuals who may have polymorphisms that may or may not result in some response. We don't know whether it's altered or adverse drug reaction, but nevertheless, it flags up these individuals so that one could use this information upfront in clinical decision making. So that's one direction that we thought could potentially help, or a direction that could be taken from some of these studies.
It's a critical question, and work
Absolutely.
from St. Jude in particular has led a lot of this, pharmacogenomic description
and already, I think the Department of Pharmacology has been leading the way in this. The previous CEO, as well as the chair, they've made phenomenal contributions, and several of their studies are used as landmark studies for many other initiatives.
Yeah. So another question now from KK Yan. So you showed a plot in which the number of expression signatures increases as the number of isoforms increases. In theory, the increase could be exponential because of combinatorial explosion, right. but the plot looked pretty linear.
That's right.
Do you have any thoughts on possibilities like selection pressure?
That's a great point. I think one real interesting mechanism about alternative splicing is every isoform expression requires potentially an RNA binding protein that will have to bind to an intron or an exon and result in skipping of an exon or inclusion of an exon. So I think it is a complex interplay between the presence of an RNA binding protein as well as the generation of a particular isoform that may serve as an alternative mechanism that can constrain the combinatorial, what you may call, explosion of possibilities that may potentially be expected. So that could be a constraint that may be operating. But apart from that, there could be many other isoforms that may be expressed, but might be potentially candidates for nonsense-mediated decay. So they may never be made into proteins, or they may actually be more rapidly degraded. So perhaps many of these combinations may actually be happening, but may be removed because of mechanisms like NMD that could remove the non-functional isoforms. The last point I think is many of the GPCRs are monoexonic, so you don't find too much variation, but the isoform combination comes from this 40% of these receptors, which do have more than 2 exons. So there are not too many receptors with 10 or 20 exons. So the combinatorial diversity of different isoform sequences may not be very high. So that could potentially contribute towards the reduced combinatorial isoform diversity that we observe.
If I can ask a question to follow up on that. So in your analysis, just thinking about associations with disease traits or cancer, in your large-scale analyses, what's your gestalt on the relative burden of association from these splicing variants to, say, more simple missense variants? And is that influenced by the nature of the receptor, whether it's monoexonic or otherwise?
Yeah, I mean, it's interesting again because there are some chemokine receptors that are monoexonic and others that are multiexonic. And just focusing a little bit on that, not us, but others have looked at how often you can tolerate deleterious mutations. And whenever you have more isoforms, you're more often likely to tolerate what you may call as a truncation, because some isoforms skip that exon. So you can have loss of function mutations to be more prevalent in genes that have multiple exons, simply because they end up generating diversity, and in the diversity, some exons that may harbor a loss of function mutation like a stop codon may still be tolerated. So I would expect that the mutation burden may be different depending on whether, but I don't have an answer per se for GPCR at the moment. Yeah.
So we have more questions, but with that, we are at the top of the hour, so we'll conclude this part of the presentation. So for those that are leaving, thank you again so much for joining. I think you'll agree this has been a really fascinating and superb presentation. But for those that wish to stay, please do, and we'll continue questions for the next up to 30 minutes. So with that, the next question is from Narav Shah. Great presentation, thank you. Very interesting work on varying isoform expression across different tissues. I'm wondering if you've looked similarly for any other group of proteins and if the computational pipeline to do this analysis is available.
Thanks, Nirav. I think this is something that I just very briefly touched upon. So we have been looking at nuclear receptors. So we wanted to look one on the membrane, one in the nucleus, and then with Charles, we also have this Alex Lemonade Foundation grant where we will focus on zinc fingers, which are another large family of proteins. Now, unfortunately, it is non-trivial to have a computational pipeline that can be relatively straightforwardly deployed at all these levels that I described, simply because one needs to understand the structure-function relationship in very great detail before embarking on interpreting some of the mutations or isoform data, because that gives rise to protein family-specific biology. So Gang Wu here, has set up this Center for Advanced Bioinformatics, where they've done amazing work in creating pipelines, and Jinghui has done amazing work on St. Jude Cloud, where you can map all the mutation data with other external data sets like post-transit modification data. So it is possible to automate at a certain level, but this is not something that we specifically do in our group. Hopefully, we can iron out some of the critical steps. Then we can make some of these approaches more readily available to others interested in specific proteins and protein families.
Great. So from Ali Salahpour, great talk. How do you plan to assess the actual physiological consequences of the various isoforms?
Hi, Ali. Yeah, sorry. Yes.
Okay. Yes. There's a second part to a question. Since these are identified in humans, I'm unsure whether heterologous cells or even animal models could work. What do you write?
Absolutely critical point, Ali. So one way by which we were really thinking of taking this further is really to do a cross-organismal comparison of isoforms, because isoform diversity itself can evolve during speciation. To be able to even know whether a mouse model is a relevant model for a certain receptor or its isoform is a useful first step. So we thought one way to move in this direction is essentially to identify, how much isoform variation, whether there are orthologous isoforms that are present between human and mouse, to even be able to identify the relevant model organism for probing the physiological relevance of some of these isoforms. So that's one possibility to take this further. The second possibility is really, again, to use epidemiological data. So I think in our presentation, I mentioned a very small part of my talk where we tried to map genetic associations, which map only to one isoform, but not the other. So these can be used to prioritize which isoforms to focus on. With so many isoforms, it's not possible for one single group to pursue this further. But with the availability of genetic data sets where you can at least have correlative associations, it's still not causal, but nevertheless, it could become like a first instance to prioritize and focus on some of these receptors or receptor isoforms to study them in greater detail.
Thanks. Next question from Prash.
Yeah.
Madan, thank you. Great talk. Are there mutations explored in open chromatin elements or enhancers which would influence the regulation of GPCRs?
Yeah, I mean, that's a very important question, Prash. So one of the MPhil students in our group initiated this project where we started looking at transcription factor binding sites. So we wanted to integrate ChIP-seq data and essentially look at their binding profiles in different tissues or cell types to ask which GPCRs are very highly regulated, what are the transcription factors that regulate a receptor of interest. So this is an ongoing project. I don't have an answer, but going back to Jinghui's question, there must be non-coding mutations that do and will influence the expression level of these receptors, especially like in chemokine receptors, where you can have people who express very high expression level. For instance, in this recent study that was published either in Lancet or New England Journal of Medicine, several key mutations that gives rise to quite serious complications upon infection with COVID ended up mapping to chemokine receptors in the non-coding regions, suggesting that they may have a role in altering the expression level of critical chemokine receptors in immune cells that can give rise to overreactive individuals to infection. So I'm sure this can contribute to natural genetic variation in the human population, and these are important questions to pursue. But having this framework, I feel, is a very interesting and a good starting point to investigate some of these questions.
And from Elaine to Armanen here at St. Jude, bacteria target GPCRs. For instance, all respiratory pathogens target PAFR with P. coli. Have you considered any infectious diseases in your data journeys?
That's a great question, Elaine. One thing we were particularly excited were with chemokine receptors and viral infection. And there's another interesting example of, I think it's, if I remember right, Yersinia pestis that also targets the beta-adrenergic receptor through a glycosylation site that was described by a colleague of mine from France. So clearly the crosstalk between interkingdom interactions between a bacteria or a virus, or metabolites secreted by bacteria, and how or the gut microbiome that could influence the expression level or signaling capacity of a receptor are all very important questions. We hope to go in this data journey by taking one protein family at a time. And we thought the chemokine receptor is an interesting protein family because we have about 23 receptors, 4 to 6 ligands. So that's a nice complex network. And that's one area where we're hoping that, we could look at how pathogens exploit this, because in the chemokine receptors, as you know, you also have viral chemokines or viral chemokine receptors. So these are decoy receptors that can squelch chemokines that are expressed by the host, or they can result in constitutive activation of a certain signaling pathway that mediates infection of these viruses. So we thought that's a very good nexus for moving in that direction of how pathogens exploit GPCRs. And the last area where we are also looking at are fungal GPCRs. So because fungi have a unique family of GPCRs, they really serve as an extremely attractive drug target, simply because they're expressed on the membrane, they are essential for fungal biology and reproduction, and GPCRs are targetable classes. So these are the two areas where we're thinking about how to integrate GPCR research into infectious diseases.
Great, thanks. If I can ask another question,
Please, yes.
Madan, as we've seen in cancer and leukemia, ligand or chemokine mutations are exceedingly rare in cancer and leukemia, whereas signaling pathway mutations writ large are often very common. So why do you think that is? Is it because they would be too broadly deleterious, and as you've shown, regulation at the receptor level constrained tissue-specific effect? Or
that's a very, very critical point, Charles. That is absolutely right. So because some of these receptors are pleiotropic, especially chemokine receptors are so relevant for development that any deleterious mutations may have developmental consequences that we may never see, simply because it may result in some sort of a lethality. But nevertheless, it is becoming clearer that more than coding mutations, non-coding mutations that alter the expression level of the receptor can make individuals, have more, I would say, primed cell motility in some cells compared to the others. So I think interesting biology may be discovered through understanding how expression level can be modulated of critical GPCRs that can, contribute to cell migration or timing of signaling, extent of signaling, as much as mutations in the protein sequence. Hopefully being able to more quantitatively measure expression level, not just at the transcript level, but also at the protein level, because you can have mutations in ubiquitination sites or mutations in 5' and 3' UDRs that can be regulated by microRNA. So these are all very interesting avenues. And Manbir Sandhu in our group has been focusing on microRNA-mediated regulation of GPCRs and their isoforms. Perhaps in the next year or so, we may have some interesting insights there. Yeah.
Great, thank you. Well, that concludes the questions. So thank you again, Madan. This has been really fascinating. And thank you again, everyone, for joining. And again, encourage you to join us again next week for the talk from Xiaomin Wang from Epidemiology and Cancer Control at St. Jude, another large-scale data science talk on genomics and epigenomics and survivorship. So we hope to see you then. Thank you, everyone, and thanks again, Madan.
Thank you, everyone. Bye-bye.